wonderful team of inverse problem makers. Two real biologists, Ailey and Paul, and my PhD students who have been working on this for quite a bit of time. So as this is not a biology talk, I actually would like to start with a little prelude, try to explain in a self-contained way what exactly the problem is. So talking about building blocks of life. So I guess probably many or most of you last time, like myself, studied biology was more or less in high school, and this is what I more or less remember from the drawings on the board. This is of course a cell, an animal cell. Somehow we imagined it as a bubble of water with some stuff, some structure inside. And actually, what I found eye-opening was this picture. So this is a more realistic depiction of what is going on inside the cell. These are the major biological molecules depicted at the right scale and right concentrations inside the cell. So you see, it's a very complicated, very crowded place. And the true heroes of this microscopic world are proteins, not the ones that you, I mean, also the ones that you eat, but basically they are both responsible for structure and function. They literally build up the things that are inside the cell, but there are also molecular machines that make chemical reactions happen, for example. And the function of a protein is dictated by its structure. So the three-dimensional structure of a protein actually is what confers its function. And biologists and physicists have been working for a long time, and many people work for a long time to try to actually see that structure with some experimental techniques. So one of the most ancient techniques that is used, of course, is you can think of this with microscopy, right? So you have some object you would like to see. Let's shine on it some light and look at the image, right? So unfortunately, it's an angstrom scale molecule. So you need angstrom scale wavelengths, and this goes into the X-ray range. And unfortunately, for X-ray, there are no good optics. We cannot build lenses for X-ray. So the only thing that remains is to do diffraction imaging. For diffraction imaging, you don't need lenses. And in order to get diffraction, you need a crystal. So it's kind of chemistry plus a lot of art, how you crystallize a protein, but protein can be made into a crystal, into a lattice, and then you shine X-ray light on it, typically from a big synchrotron ring, and you see diffraction patterns on it. So these diffraction patterns will actually be two-dimensional slices of projection of the Fourier transform, but you don't image the phase. You only see the absolute value squared of the Fourier transform. Because the function that we are imaging, the physical property that we are imaging is the electron density. It is periodic on the crystal lattice. The Fourier transform will be discrete on the reciprocal, right? So I'm not going to talk about recovering the phase. It's a complicated problem, but let's assume that what we start with is this reconstructed electron density. What biologists want to look at is this atomic model. Basically, we want to place in space dots where the atoms are and connect them with sticks, which represent the valence bonds between the atoms. And of course, the electrons in one atom are as good as electrons in the other atom, so they don't come with labels. We don't know which electron belongs to what. So it's a complicated problem. It's an inverse problem that it needs to be solved in order to get the molecular model. So typically in crystallographic structure modeling, our input is the observed electron density. Suppose we recover the phase, and every atom will be modeled as a little Gauss, and we will place the location parameter, and then the density, of course, will be blurred around it. So we will also fit some kind of uncertainty parameter as well, variance that the crystallographers call B factor. And then the conformation of a protein will be a Gaussian mixture model. Okay. So that now, once we have this model, we can model the forward model. We can say each atom will deposit some amount of electron density in space. This kind of expansion into 5 Gaussians with the coefficients that have been experimentally determined probably before I was born. And you can think of this just as a kind of a kernel density estimation. So we go with each atom location and we place a little blob of density in space. And this is called the calculated electron density, FC. And the purpose of modeling, of course, is to make a match, make FC match to the
Why 5?
Well, it just described well the experimental.
Okay. It's not related to
No
number
fundamental reason. And of course, it also depends on this B factor, the mean uncertainty.
So does this forward model also depend on the bonds or just where the atoms are?
Yeah, of course. So the atoms, the chemical role of the atom affects these coefficients. Like for example, carbon, C alpha and C beta and C will have different.
Oh, but maybe I asked my question wrong. But the lines between the dots, do they show up in this formula, or is it just the dots?
No, just the dots, and the lines can be inferred. Just
okay.
If the distance is shorter than some number, then they must be covalent.
Okay.
But basically, we lose a lot in such representation. So if you go to the protein data bank, to the PDB, and you open any structure, you will see this representation. Actually, the sticks are not even shown because they are inferred. Essentially, we see a mixture of marginal distributions of the atoms. All the information about the joint distribution is lost. And the joint distribution is non-trivial. You have these little balls connected by rigid sticks. The angles typically between every 3 balls they are rigid. They're basically dictated by the chemistry. The hydral angles are non-rigid. So all this information is lost. If you just try to sample independently each atom location from this distribution, you will get nonsense, completely non-physical model. And why is it important? Because seeing the protein, like I showed before, is a big lie. The protein is actually a dynamic structure. So this is the molecular dynamic simulation of one of the SARS-CoV proteins, the spike protein. So you see, it moves, it oscillates, it has dynamics, and what we actually observe in the electron density as blur is not because you cannot get a good signal. That is the signal. That is part of the physical reality of the protein. It is blurred because it moves. We want actually to use that information. It's important. So basically, the electron density is an ensemble average. You freeze about 1 quadrillion molecules in liquid nitrogen. They stop moving practically, and each of them is captured at a different conformation. So what we see is an ensemble average on this huge amount of molecules of this distribution. Now, talking about another experimental technique that is very well attuned for getting dynamic properties of proteins is nuclear magnetic resonance spectroscopy, or NMR. So NMR is based on a nuclear phenomenon. It's a nuclear phenomenon based on the fact that some spins, some nuclei have a property that is called spin. When you place them in strong magnetic field, they will align with the direction of the field. And then if you excite them with radio frequency that matches a very specific resonant frequency, they will rotate and rotate back and emit that radio frequency back, and we can detect it with an antenna. Now, this is also the principle underlying MR imaging, but in MR imaging, we have another magnet that creates slight variations of the magnetic field in space. So different locations in space will resonate on different frequencies, and this is how an image is formed. Now, of course, MR people would die for having this kind of spatial encoding, but for molecular size, you will need probably the magnetic field of a neutron star to create such a gradient. So no spatial encoding, all the peaks are completely mixed. However, even though this is a nuclear phenomenon, the nucleus is surrounded by electrons, and electrons interact with the field. And depending on the chemical role of the atom, how exactly the geometry of the bonds is shaped, the electron orbitals will deform and will shield the nucleus from the external field by a different degree, and we will see slightly different resonance frequency. And this is all the signal that NMR people measure is there in those ppm scale chemical shifts. So the chemistry affects the resonant frequency. They see a soup of peaks, thousands of peaks coming from a typical protein molecule. And how the heck do you assign these peaks to atoms? So what they know how to do is actually a very clever technique. You can measure correlated peaks. You can excite one nucleus, of course, multiple nuclei will be excited at the same time, and transfer magnetization to the neighboring nucleus. So then you see a peak in this two-dimensional space, carbon and nitrogen showing a two-dimensional peak. Then you can do the same for the overlapping nitrogen and carbon. So you know that it needs to be the same nitrogen, so it needs to share the same coordinate for the peak. And then you go like this, you basically stitch up this puzzle. And if you have ambiguities, you need to go to higher dimensional correlations. It's an amazing correspondence problem. Next time, if you invite me, I will show you some completely out of the box attempts to solve it using different tools.
What's the difference between nitrogen and nitrogen and carbon?
Well,
On the graph on the right.
here.
Yeah.
Well, this is just a visualization, but so all these grays are carbons. They have different chemical roles. They have different chemical shifts. But you might have many other carbons in a big protein that have very similar locations.
What happens when you correlate carbon and nitrogen with carbon and nitrogen? I mean, what's the difference between the two graphs?
Oh, well, so it's a pair of carbon and nitrogen. Here is nitrogen and carbon. So they share the same atom. So these coordinates must be shared.
So it should be the other way.
You need to have overlaps.
So how do you excite the left carbon without exciting the right carbon? Like,
Well, that requires more than the 5 minutes.
okay.
I can tell you. So, and very important kind of experiment that NMR people do for determining protein structure is based on what is called the nuclear Oberhauser effect. For example, if you look at these two hydrogens, they will form some peaks. You can, using the previous procedure, you can assign them to specific atoms. But then magnetization will also transfer through space. So if they are close enough, you will see a cross peak in this correlation domain. And the intensity of the peak will depend on some inverse sixth power of the distance between the atoms. And actually, just remembering that the protein is not this static picture, but something that moves all the time, it will be actually a temporal average of that. So essentially, we can measure distance information in a protein. Right? So reconstructing structure is an interesting problem. You reconstruct shape from distance. Okay, so this is the kind of things that NMR people can measure, and they can measure many other things, but everything they measure is some kind of temporal averages with different temporal scales. Now, another thing that I want to mention, and we will be done with this introduction, it's important also to remember how proteins are created, how cell manufactures proteins. So just a brief mentioning of biology. So first of all, the coding instructions for the protein are stored in the DNA that makes up the genome of a living organism. A gene is actually a portion of the DNA that codes for the protein. The first step is called transcription. This DNA molecule, which is very stable, is some kind of long-term storage, is transcribed into a very perishable molecule that is called messenger RNA. It's unstable. It lives for short enough time to go to this amazing molecular machine that is called the ribosome that knows how to read those instructions and create a chain of connected amino acids. This is what makes up the protein. And the final step of this play is by electrostatic interactions and thermodynamic processes, the protein folds into a three-dimensional shape. Okay, so this is how it looks like. This is about 4 times faster than it happens in reality. I think this is just amazing. The protein, of course, starts folding before it is completed. And one of the holy grail problems of structural biology was how do you predict how the protein will fold? In theory, if you know the amino acid sequence, you know how the shape is going to be. But of course, you need to solve, you would try solving Schrödinger's equation, for example, but this is so much out of scale of any existing or any future computation power that we have that it is completely out of question. People try to do it with some kind of principled ab initio solvers quite unsuccessfully. And the groundbreaking result came about a decade ago and got the Nobel Prize for chemistry in the past year, AlphaFold. So it's a machine learning problem. You can think of this as a machine learning problem, and it's a machine learning solution to the problem of predicting protein structure from the sequence. So basically, you have sequence, give me the structures. There is in the protein data bank probably about 250,000 useful sequence structure pairs. Sequence structure pairs, right? So you can say, I want to find this relation. I want to generalize beyond what I see. We have these 250,000 annotated or labeled samples. Let's just learn this neural network to predict structure from sequence. But you can do it and you will fail. Won't work. The sample size is tiny for the complexity of the relation that we need to discover. So this is not how AlphaFold works. Actually, AlphaFold works because of a discovery that was made in the 90s by Sander and other people for who surprisingly didn't get the Nobel Prize. But basically what they discovered is this. If you take a sequence of amino acids, this sequence will also appear with some variations in other organisms because life reuses the same building blocks, they evolve, they will undergo some mutations. So you can bring from an external data set of sequences, similar sequences and align them together, right? And then what they discovered is that there will be pairs of locations that mutate in a correlated way. And what they showed actually is that those pairs of locations correspond to spatially proximal. So if you change this amino acid, you also need to change its interacting partner, otherwise protein maybe will break up. So from that, you can infer some quantitative map of spatial contexts, and learning from that input the structure of the protein is a much more tractable machine learning problem that indeed AlphaFold solves. Essentially, it has this multiple sequence alignment stage. Then it has this pair formal network that creates an embedding for MSA. And these conditions, at least in AlphaFold 3, it's the diffusion model that generates the structure. So basically, it's the availability of cheap DNA sequencing that was made possible in the 90s that created hundreds of millions of sequence data points, most of them without structure, of course. Basically, anything that lives on Earth more or less has been sequenced. And from that, we get the structures. This is how AlphaFold works. But remember, it has been trained on those data that have been generated in the protein data bank over the course of many decades by algorithms that are, in the best case, 20 years old. And they represent protein as this kind of blatantly wrong picture, right? So AlphaFold is as good as this data. Okay? So what we would like to do is, this is the main topic that I wanted to show you today, is how to fix that inconsistency. So it's kind of an unhealthy situation. We want to solve an inverse problem. We observe some experimental observable. We want to solve for the protein structure for this atomic model. We want to capture all the dynamic properties that are part of the protein reality. But we also want to use those very strong evolutionary priors that AlphaFold exploits. But AlphaFold, of course, will have its shortcomings because of the data it was trained. So first of all, I would like to start with proposing a different representation. So in the traditional crystallographic community, this is how the proteins are represented, right, as mixtures of marginal distributions of atoms. That, of course, completely loses sight of the joint distribution. So what I would like to do instead is to think of, I want to have a black box from which I can sample an ensemble of structures. So each element in the ensemble will be a realization from the joint distribution. I want to condition the joint distribution by the sequence that I know, and also the experimental observation, whatever it is, with crystallographic electron density or NMR distance constraints or something else. Okay, so I want to sample from this posterior distribution. Now, sampling from this posterior, of course, can be factorized into the likelihood of my experimental observation, like the electron density given the ensemble, times the prior, which is basically what I know given the sequence of amino. And the prior, of course, is separable. Every element in the ensemble, in the absence of any other information than the sequence, I can predict it independently. The likelihood will typically be inseparable. So let me show why. Let me show you two likelihoods. For example, in the case of X-ray crystallography, it's just the forward model of our instrument, right? So I have an ensemble of structures, and I'm asking what is the probability of the observed electron density, right? So I need to somehow measure the discrepancy, quantify the discrepancy between the FO and the calculated electron density. And the calculated electron density will be just the ensemble average over the calculated electron densities. Remember, with those little kernels, 5 Gaussians that deposit density in space. So I need all the members of the ensemble to calculate this. It's an ensemble average. And then I need to compare it to the experimental observation. Therefore, I cannot split this term with respect to the ensemble member. Same story for the NOI, for the nuclear Oberhauser effect, for the NMR observable. Typically, it comes in the form of not exact distance measurements, but upper and lower bounds on some collection of sparse collection of pairs of atoms, typically hydrogens. So let's say I have these two hydrogens. I know that the distance between them is above some lower bound and below some upper bound. Okay, and again, the distance is an ensemble average. So I need to average my ensemble again. That's why it is inseparable, and compare to the bound. Okay. And so that was the likelihood. And of course, if you have another physical process, another instrument, you can put another likelihood term. If you know how to model the physics, then you can put it into the model. For the prior, we would like to use AlphaFold's diffusion model. Right? So we want to sample from, again, it's still a conditional distribution, but now only conditioned by the sequence. So the way diffusion models work, and I will just briefly mention this, we would like to sample from some complicated distribution, right? What normalizing flows or diffusion models in particular do, they create a trajectory, a path in the space of probability distribution. So we start with some trivial distribution from which we can sample, let's say a Gaussian distribution. And then we flow our samples by a sequence of, or a continuous flow, basically nonlinear transformation that will, by trivial change of variables, will transform our distribution into the desired shape. So typically, these models are trained by starting with the data, going backwards by adding noise. So we blur them, we convolve the marginal distribution with a Gaussian until it becomes Gaussian. And this is, of course, described by a trivial diffusion process. What is surprising and quite beautiful is that the reverse process is also a diffusion process. So it can be written by this stochastic differential equation. And everything that describes the probability path goes into the score function that drives the flow, right? And of course, that part, that's the core of the model. It is typically represented as a neural network. This is what is trained in the AlphaFold structure. Okay. Now we can of course generalize it to sampling ensembles. So this is an ensemble of n elements. Of course, this is just running n diffusion equations in parallel. They are completely independent. Now, if we want to sample from the conditional distribution, if we want to add another conditioning by an experimental observation, we need to replace this with a conditional score, right? But the Bayes theorem is very cool for score because it has log and it has gradient. It will just have two terms, right? One term is this unconditional score, so continue sampling unconditionally, and another term will be this gradient of the log likelihood given the experimental observations, right? And this is the inseparable part. This is what couples the different diffusion equations together.
It's just like the conditional guidance.
Yeah, this is conditional guidance, except that it is a very, very weird guidance because it's non-IAD. So the ensemble that you are sample, the members of the ensemble are depend on each other. Typically, conditional guidance is done in an IAD setting, right? And this is kind of strange non-IAD guidance.
But usually you don't end up with posterior distribution if you just add. So usually what you would need would be somehow the condition of your current time of your diffusion path at the current time, right?
Yes. So of course. So I'm omitting some details, but basically those x's depend on time, right? So you need to evaluate this likelihood on the noisy sample as well. So typically it is done by doing some denoising step and then evaluating the
Yes, but usually, I mean, if you have the likelihood for the sample at the finite time, and then you blur the samples, then the likelihood of the blurred samples. So I'm.
Yeah, no, I think we're talking about the same. So you need to evaluate. So the x's at the final time, right? When you reach the final diffusion time, this is supposed to come from your conditional distribution, right? And the likelihood will exactly evaluate that. During the diffusion, right, where these x's are noisy, right? So you're basically putting some noisy samples from noisy distribution into your likelihood. You need to denoise them first. There is some magic, some details that need to be done correctly in order for this to work well. But basically, what this red term does is biases the unconditional diffusion towards your conditional distribution. Okay. So basically, this is how the entire thing works. This is the original AlphaFold. It generates an ensemble of unconditionally, it would generate just a trivial ensemble, typically a very small variability, an ensemble of structures. Then we calculate ensemble statistics, for example, the electron density or the ensemble average distances. So this is where our experimental inputs come from, right? From X-ray crystallography or from NMR. In crystallography, typically, we also want to refine some part of the structure. So if we want to refine a part, we want to fix some of the atoms in place. And then it would be some kind of a separable likelihood term that freezes those atoms or penalizes for deviation from some target locations in every member of them. So then it wouldn't be an ensemble property, it would be an individual ensemble member property. And we use this to guide the diffusion model. So essentially, we both use the experimental observables and the biological input that comes from the multiple sequence AlphaFold.
So, and I have a question. So AlphaFold never used the electromagnetism or the
AlphaFold is trained on structures that were reconstructed from this data, of course, right?
okay.
But it's kind of an unhealthy situation, right? You reconstruct your structure with a poor algorithm, and then you train a model with that algorithm, right? And then you need to fix it with this guidance to actually match the experiment.
Well, if you have enough examples, then
Well,
you
of course, but don't steal my time. So basically, we have these guidance. I will show you some examples. I think I'm doing okay with the time. So some examples. So this is, for example, this is the SARS-CoV protein from the 8th opening reading frame. It is implicated in the virus's ability to evade the immune system. But basically, there have been some crystallographic structures that appeared surprisingly within a few weeks from the virus outbreak, and they show quite significant conformation heterogeneity in 3 regions, even though, for example, these 2 proteins here, these 2 structures differ only by 1 mutation, which should be inconsequential for this structure. But of course, AlphaFold will see the same MSA and it will predict the same result, and indeed it does. So here are these dark green and dark purple. These are the experimentally determined structures. They have very clear electron density, pretty well fit. But AlphaFold either predicts one of these variants or something else, right? So it completely misses. And of course, there are tons of examples like this that AlphaFold doesn't match the experiment well. If we use guidance, we actually describe the electron density very well. So this is the case that AlphaFold fails. I unfortunately don't see the dark well, but these are basically the dark green here and the dark purple here. These are AlphaFold predictions. They stick out of the density, they don't describe it well. And the ensembles generated by the guided AlphaFold, these are the light green and the light purple, they actually stay within the density and describe it well. And here, for example, we also see some additional conformation heterogeneity, some additional modes that were not captured well by for example, the original modeling in the protein data bank. This is a more dramatic example. Actually, this is a very interesting phenomenon. So this is one crystal, one single protein that harbors 2 conformations of the backbone, and actually very distinct conformations. So one forms this one loop of a helix, another one doesn't. They are very distinct, clearly no mistake. Actually, this protein comes with 4 chains. It's a tetramer. All chains have this. So it's not something that can be attributed to some weird interactions. This is actually the nature of this protein. It has bimodal distribution of the backbone. It might be switching between the 2 modes at some time scale. We don't know that. We cannot determine the time scale from crystallography. But surprisingly, this phenomenon has been largely overlooked. We did some large-scale study on the PDB. About 15% of structures exhibit this kind of behavior. AlphaFold typically will predict just one of them. And actually, AlphaFold was trained. So sometimes in the PDB, this was actually modeled in the protein data bank. They actually bothered to commit 2 variants to the protein data bank. Most visualization software and AlphaFold training set just takes the first one, which is incredible, I think. So basically, you never observe the second one. If we guide by the electron density, we actually are able to recover both structures. Of course, explains the electron density much better. So you see the calculated electron density here with this mesh. It misses half of the density here, and we get quite a good match as well. I have quantitative data, but I won't bother you with tables. Yes, please.
Just to make sure I understand it correctly, the way you explained AlphaFold, there is a diffusion component, so it can be a generative model.
Yes.
If you retrain AlphaFold with taking both or multiple ones instead of the top, would it?
Yeah, I think I would like to retrain. Of course, so what I have a slide on that, but let me anticipate. So what we are showing here is essentially some kind of transductive mode, right? So I'm observing a single experimental measurable, and I want to match that. But let's take a collection of those experimental measurements, corresponding sequences, and train AlphaFold directly on those measurements, not on something that has been reconstructed, fitted into them, but directly on the measurement. Match me my electron density, match me my NMR restraints. So that would be an inductive model. This is something that we are currently actively working on with quite promising results, but I still don't have anything good to show you. Of course, that's the better way, right? Because you want to solve an inverse problem, you want to generalize beyond your training software, right? But here it's entirely transductive. You observe this electron density, give me an ensemble that models it well. It's not only, but it's important not only to estimate these two conformations, but actually what is the probability of being in each of them. That is, for example, crucial for enzymes that there the flexibility can alter the enzymatic activity by each factor. This is just a visualization of more proteins that exhibit this kind of bimodal behavior. What we are showing is that when we do guidance, this non-AD guidance that I described, you actually get this multimodal behavior very well. Here, basically, this kind of weird sign distance tells you how close you are to the two conformations, and when the AlphaFold is not guided, typically check one of them. I would like to show you also the solution of this inverse problem of reconstructing the structure from distance constraints that we measure with NMR from nuclear overpowers effect. This is the NMR structure that was deposited to the protein data bank. So the way NMR people typically solve for such structures, right? So there are about 15,000 structures deposited in the PDB reconstructed from knowing constraint. So what people do, they run molecular dynamics simulations, which has the electrostatic potentials modeled. So you get an ensemble, you can get basically a trajectory or an ensemble of structures, and they add on top of it some kind of a fake potential that penalizes for violation of the distance constraint. This is very expensive. MD can run for days, and you don't get non-IID samples. So basically, you impose distance constraints on that specific run, and then you run multiple times and sort the results by energy, and you typically get tight ensembles, not very descriptive. So here, for example, this result, I think this is ubiquitin, about 10% constraints are violated. Among those violated constraints, about 0.3 angstrom median violation. AlphaFold gets it really wrong. So 1/4 of the constraints is violated. Also, the violation is quite strong. You see basically all those violated distance here with the color bar. And when we guide AlphaFold with distance constraints, we actually get better results. So in many cases, we get better results than what people obtain with molecular dynamics, with the exception that this runs in 2 minutes, right? And actually, with this time complexity, we can redo all the PDB structures from room, right? It's completely impossible with molecular dynamics simulation. So this is an example of a few ensembles. The Y structures is what exists in the protein data bank. You can see that AlphaFold often completely misses the correct tertiary structure of the protein. So here, for example, it went some completely wrong direction. And when guided by knowing constraints, it actually matches quite well the white ensembles, but numerically it often performs better. Another interesting thing that some kind of additional validation, NMR can measure many cool things. One of these things is the distribution of the direction of different bond vectors. For example, this is a very convenient bond because you can isolate it very well in NMR experiments. This is the amide bond, nitrogen to hydrogen in the backbone. Right? So every backbone has a nitrogen and the little hydrogen that sticks out of it. You can actually look for this unit vector in the direction of this bond, and with NMR measure basically some temporal average of these correlations of this angle. And typically, you would expect that flexible regions in the protein would have bigger variations, so you will see this S squared parameter will go down, and more rigid places will stay rigid. So indeed, if you look for ensembles predicted by AlphaFold unguided, you get very flat plot here compared to the experimental observation that indeed highlights low values for flexible regions and high values for inflexible regions. For example, this is a helix; it's a very rigid structure. So you have high value for this order parameter, and for flexible regions we have lower values. So if you correlate the ensemble estimate of this parameter versus the experimentally measured one, you actually get very poor correlation for AlphaFold. When guiding AlphaFold, we get significantly higher correlation. So here we also tried to compare IAD guidance versus non-IAD guidance. So in IAD guidance, we can just require every member in the ensemble to satisfy the restraint. And this is physically wrong, right? Because what we measure is actually an ensemble average. We get better correlation, but still quite low. So this non-IAD guidance is a very important step to improve the agreement with the experiment. And actually, you cannot really do it in MD. I mean, in theory, it is possible, but it will really take weeks to calculate. And here it takes 2 minutes to compute. So this is just an example. 2 lysines in position 11, position 29. This is the flexible region. This is the rigid region. You can see that AlphaFold more or less predicts them the same. And if you look at guided ensembles in the flexible region, you see this kind of mess, very spread, the directions of this bond, and in the helical region, it is less spread. So just to finish, and almost done. Yes. So a few next directions that I'm really enthusiastic about. So one is the new tool or old new tool that is coming to structural biology for probably a decade already. There has been what is called the resolution revolution in this technique. I'm talking about cryo-electron. So unlike photons, electrons are charged particles. We know, well, not we, but some other guys know how to build lenses for electrons very well. So you can actually do real imaging. And when you do imaging with electrons, you actually observe individual particles, individual molecules. This is the scenario. It's about 0.1. I don't know, do you see a protein here? It's a projection. It's a 2-dimensional projection of a 3-dimensional property that is called the electrostatic potential. Electrostatic potential, which is kind of electron density, but it also involves the nucleus. So you would like to reconstruct this volumetric image, but remember that this volumetric image, what you see, there is a flexible molecule underlying this, right? And what you see here is an individual realization of one of its conformations that has been frozen in liquid nitrogen, but you see only a projection. So basically, with each such image, you can associate 2 latent quantities. One is the conformation, which is different in every realization, of course, and the other one is the orientation, which is also unknown, right? Because you just see randomly oriented. What is nice is that you can get, if you're lucky, 500,000 such images. So it's a lot, and that is to compensate for the poor scenario. So I would like to go directly without reconstructing the density. I don't care about the density. I want to get the atoms and samples of atoms. I would like to get the distribution, the joint distribution, conditioned directly by this. Okay, so I think this will currently you first reconstruct the density without taking any structural priors, and then you try to fit the structure. So that gap looks like something that needs to be bridged. Another thing that, of course, has been raised here several times. So currently, we are operating in this transductive regime. You give me a specific sequence, a specific experimental measurement you want to get the ensemble of structures.
Alex, just a second. Regarding the previous one, this is something that you would like to have, or you're preparing for an answer for what you're now?
No, we're actively working.
Okay.
I mean, structural
So
biology has been working on this. Conformation heterogeneity is currently a huge problem that they don't.
It's like INR or NERF without knowing the directions.
Well, people started using NERFs for representing density, but I want to keep the density stage altogether because I'm not interested in that. Right? Yeah.
But right, if you use the volumetric rendering from graphics, it would not quite apply here because graphics do huge shapes in.
No, I agree. It's not. No, it's.
There's fluid and other interactions.
Yeah, no, I agree. But you can model the forward model quite well. So you have the likelihood. So what we have been doing now is transductive. But what we could do is we have a collection of experimental measurements, electron densities, NMR observables, cryo-EM observables. We have them associated with the sequences, right? Why don't we learn AlphaFold directly on these data? I don't want to first fit a structure into this data because this is the problem that I would like to solve in the first place. And then I wanted to generalize. Then given a sequence that I didn't have in the training set, I would like to get an ensemble that would explain the experimental observable if I take that experimental observable, right? So this is what I would like, where I would like to get. Of course, it is computationally much more expensive to train a big model and such data, but not prohibitive. So with this, I will finish. We can look at a few papers on this on these topics on my website.
Yeah, so at the start of your talk, you showed an animation of a protein moving, right? And so my understanding is that the conformation of a protein in solution can be quite different from what it looks like when crystallized.
Yes.
And right now you're guiding based on the crystallized structure, right? Is there some way to also learn the shift in distribution due to crystallization and take that out?
Yes, it's a great question. So first of all, there are some level of dynamics that match well in solution. So people try to do solution and solid state NMR with crystals. There are some dynamics that remain the same. Some bigger and longer time dynamics change by orders of magnitude. So of course, when you look at the crystal, things change, right? It's not the most natural environment. In that sense, what you actually see in the electron density is not exactly the Boltzmann distribution. It's constrained by the crystal complex and so on. That's why NMR and cryo-EM are so good because you actually get to see your protein maybe even without purification, just in its natural environment, right? So this is why it is a much better experimental technique. So doing this kind of attempt to translate properties from crystal to non-crystal, you need some significant amount of data, probably coming from cryo-EM to actually make it work, right? You need significant amounts of data. Cryo-EM still doesn't reach that level of resolution that crystallography does, but cryo-EM is very good at imaging complexes, imaging interactions with ligands. So that is something that it is very good for. So another thing that AlphaFold 3 allows for is actually having more than one molecule. And you can sample from the joint distribution of, let's say, a protein and ligand. That is something that is not modeled well in existing DB strategy.
Maybe we should.
Just before the second talk, there's one thing that we forgot to mention. All the talks that we recorded, the world records all those talks and then puts them onto the internal website. So